Off, it's my pleasure to introduce our first speakers, Vishal Shah and Shefali Thanawala from Nutriventia Private Limited. They will be discussing redesigning nutraceutical efficacy and how Turma Extra and Prolons deliver measurable health gains in small doses. Let's please give them a warm welcome.
Good afternoon, everyone. It's truly a pleasure to be here amongst fellow innovators and leaders of the natural product industry. I want to just spend a little time and talk to you all about the genesis of Nutriventia, how we all started. So we were an offshoot of Inventia Healthcare. That's a pharmaceutical company which is a pioneer in novel delivery systems. And we've been at it for close to 40 years. The thought behind Nutriventia was very simple. What if we can bring the same level of innovation which we see in the pharmaceutical space all the way up to natural products? When we look very closely, one of the things that we noticed is that a lot of companies were talking about standardization and extraction, but there was no one really talking about the delivery aspect. How do these natural ingredients actually behave inside the human body? And that is where we started, and that's how all our products are designed. And love to kind of walk you all through this entire journey. So these are some of the platform technologies that we have developed over the last 5 years. Everything from enhancing bioavailability and lowering the dose, modified as well as time release of actives to improve compliance, site-specific release of the actives to enhance absorption, and taste masking to make bitter actives more palatable. The end goal for us is to make natural products more effective and easy to consume for the end users. And we do it all with clean label and food compliant excipients. So here are some of the products that we have developed over the last couple of years. So these are the ones which we have invested in a lot of clinical validation. And as you all can see, the common aspect amongst all of them is that it is a low dose and an effective 1-a-day consumption. And that's something which we really believe in, and that's what we strive for. As you all can see from the slide, we've had a busy last 5 years. We love what we do, and we would love to partner with everyone out here to spread the message forward. So without much ado, I'll pass it on to Shefali, and she will walk you all through the entire clinical journey for Term Extra and Prolanza.
Yeah, thanks, Vishal. And hello, everyone. Let me take you all today through a journey which we took up for designing, developing, and also confirming the efficacy of 2 of our ingredients. But how we did it was by using 2 seemingly different scientific principles. And these are translational research and reverse pharmacology. Well, I think I don't need to talk much about translational research. We have heard a lot about it in the morning. And I was really glad that people are talking about translational research and its importance. To put it in a nutshell, translational research is like taking laboratory discoveries to from in vitro and in vivo studies to clinical trials, population studies, epidemiological studies to develop the clinical tools for improvement of human health as put by NCAS in IH. And translational research, as we all know, is a very time-intense activity. And I can say that it's a path well trodden, and it is used very regularly and commonly in the pharmaceutical research. But when it comes to nutraceuticals, the same well-trodden path becomes a road less traveled. I think most of the nutraceutical people do not probably see the need to go into this kind of in-depth research at every step of the journey. So the natural question is, then how do we generate evidence at every step when we are designing, developing, and validating the ingredients and the products? So the answer is in reverse pharmacology. Basically, reverse pharmacology was I can say, reconceptualized by renowned physician of modern medicine from India, who was also an eminent scholar of Ayurveda, Dr. Ashok D.B. Vaidya, and I would like to pay my homage today on this stage to him. He taught us and conceptualized, and basically he told us that there are thousands of leads available in the ancient scriptures of Indian medical science, that is Ayurveda, and in spite of discarding what has been like centuries of clinical wisdom, if we use those leads, we take it back from bedside to bench. We take those leads to, again, in vitro and in vivo. Again, we take it to clinical trials, and then we present in a better, reconfirmed, validated form for users and consumers. So that is the reverse pharmacology concept, and we have used these diverse principles and converged it, amalgamated it to create two flagship brands, and the first is Prolanza. I can say it's the world's first ever sustained release ashwagandha product. And let me take you all through the journey of Prolanza. Now today some of you at least might be wondering why I'm here in this dress with a bindi on my forehead. So because I've got something for you all from India. So as you can see here, I have taken one Sanskrit verse from the ancient Ayurveda scripture, and just, these are 2 lines, right? So in just 2 lines, the ancient scholars have described the organoleptic properties of ashwagandha and also the multiple health benefits that the herb gives to us. Now, I think everybody is aware of ashwagandha, right? But how many of you really think that ashwagandha means smelling like a horse? Can I have show of hands? Oh, there are some people. Nonetheless, and I think most of you came to know today that ashwagandha actually means something smelling like a horse, and that's the organoleptic property of the raw root, dry root. However, let me tell you, this Sanskrit is a very complex language, and it has a fun element to it. Almost every word which is written in Sanskrit has a deeper meaning to it. So when the ancient Ayurveda scholars wanted to name this herb as ashwagandha, of course they meant the organoleptic property, but also they wanted to tell us that consumption of this herb will give you all the qualities which are core attributes of a horse: energy, vigor, vitality, being adaptogenic to environmental changes, muscle mass, etc. So that's ashwagandha. Now, I have been using ashwagandha root extract and root powder as a Ayurveda clinician in my more than 3 decades of practice, and I've been using it in multiple daily doses, and it gives fantastic results as an adaptogen and in multiple health issues like stress, anxiety, sleep issues, etc. Ayurveda also recommends very clearly the plant part which is to be used, and for ashwagandha, Ayurveda has clearly said that you should use only roots for internal consumption and other parts for topical application if and when required. So I have been using the root powder and root extract in multiple daily doses to get the optimum benefits, and that was the research question which we took up when we sat together in Nutriventia and looked at ashwagandha. Hey, can we really reduce the dose? Can we increase the patient compliance, the user compliance, while still retaining all the benefits that the herb gives? And with that question in mind, we started looking at the PubMed database and the available research. At that point in time, around 900 research papers were available, rather the results were available with the keyword Vidania somnifera, which has now doubled, almost doubled in past 7-8 years. But we wanted to go for dose reduction. So with that question in mind, we started looking at if at all there is anything available in terms of a pharmacokinetic data or what are even the actives of ashwagandha identified. Has anybody tried to estimate it in vivo system? And we could see only 2 in vivo PK studies. So with no documented bioavailability issues, with just 2 basic pharmacokinetic studies available, showing a possibility of a fast elimination of the actives, and with the question of multiple daily doses being to be converted into a single daily dose, we took up the challenge and developed the world's first sustained release ashwagandha product. And after taking the cue from the ancient scriptures through reverse pharmacology, we started our journey of translational research, and we conducted in vivo study. Because there was a lot of pharmacological data available for ashwagandha, we directly jumped in to take up the in vivo pharmacokinetic study, and Prolensa could show in rat plasma double the concentration as compared to the early market entrant root extract product, which we used as a reference. And Prolensa showed 11 hours of elimination half-life, and we passed our first test. The design of sustained release, which was developed by our R&D experts, replicated the results in the in vivo system. And further, because it was a sustained release product, we wanted to re-evaluate its pharmacodynamics, and we took up the study in chronic unpredictable stress model. As it is mentioned here, Prolensa showed anti-stress effect and memory retention effects in Morris-Water maze test, and sorry, I'm not able to read here. So in the other test that is mentioned here, and we got very good results in the chronic unpredictable stress model. We also evaluated and confirmed the safety of the product through entire tox pack. And the mechanistic studies, we showed the anti-inflammatory and antioxidant effect, and the journey continues. We recently conducted a neuroprotective study in the zebrafish, and this was an academia collaboration. So next was a major step for us for the translational research. There was no human pharmacokinetic data available for the ashwagandha actives, and we standardized our product for USP with danolides, and now we decided to evaluate whether the sustained release profile is getting reflected in healthy human volunteers. And with that, we designed a human pharmacokinetic study, and as you can see in the graph, there is a huge difference in the area under the curve, in the concentration, and even in the elimination profile of Prolensa as compared to the same early market entrant ashwagandha root product. The green line is Prolensa. And with that, we could see that the ashwagandha actives are remaining in plasma for 24 hours with just a single daily single dose. And with that, we conducted further 2 randomized controlled, placebo-controlled trials for Prolensa, and the results are here. In the first study, again, we took as a primary endpoint neuropsychological battery, that is CANTAB. Now, CANTAB is used very routinely in pharmaceutical research, in psychotropic drug trials and all. But when we conducted the trial, we could not find any nutraceutical having used CANTAB. So we could see great results for Prolensa in terms of memory retention in the paired associate learning parameter of CANTAB. And of course, the anti-stress effect, the improvement in sleep quality, and also the improvement in the psychological well-being, all evaluated through validated questionnaires. Prolensa performed very well during the treatment period, and even the objective biomarker serum cortisol was in sync with the results of the subjective questionnaires, and the cortisol levels dropped significantly at the end of the treatment period. Then we conducted the second trial, and we asked another question to ourselves: will Prolensa perform in half dose? And that too just given once a day. So in this trial, we had 3 arms, the 300 milligram, 150 milligram, and placebo. Again, we evaluated the same parameters of perceived stress, psychological well-being, sleep quality parameters, and we added here to evaluate if Prolensa would have any effect on the eating behavior in the stressed volunteers. As we all know, our eating goes totally haywire when we are under stress. So in the 3-factor eating questionnaire, Prolensa 150 milligram and 300 milligram performed significantly better. Interestingly, in this study, we could see that Prolensa in the dose of 300 milligrams started giving significantly superior results as early as day 7, and 150 milligram followed the higher dose and started giving significantly better results just in, from 15th day of the treatment period. So this is in a nutshell the story of Prolensa. We started from Ayurveda, we took it forward to mechanistic in vitro studies, we took it to in vivo PK/PD studies, first in human pharmacokinetic studies to confirm the sustained release profile, and the clinical trials. and then bringing it back to the users and the consumers. Let me take you to another success story, that is Term Extra. Again, turmeric is very deeply rooted in Ayurveda, extremely well praised, but we'll come here later. So, when we started looking at turmeric, the first research question, again, turmeric is used in Ayurveda, we use it in multiple daily doses because so here, why we have to use it in multiple daily doses, that question was answered because a lot of peer-reviewed literature was available showing a poor absorption, metabolism into inactive metabolites, and a fast elimination, all leading to poor bioavailability of turmeric and curcumin. So, with that information in hand, our R&D scientists put their heads together and designed and developed a water-dispersible turmeric extract with 60% of curcuminoids. So what happens is in market when turmeric is, when the scientists start improving bioavailability of turmeric and curcumin, the active component goes very down. So we have the products generally in market starting from 10% to 20% of actives. So our scientists developed, probably it is again for the only product with such a high curcuminoid content. Because there is lot of data available on the multiple pharmacological action of turmeric and curcumin, we all know it's a pleiotropic molecule, right? So we directly jumped into an in vivo pharmacokinetic study, and at the same dosage, Term Extra showed 10 times improved bioavailability. Taking cue from this, we conducted not one but two pharmacokinetic studies in healthy subjects. Now the question will be why two? We all know that turmeric is a very variable ingredient, and its plasma levels vary extremely, there's extremely high variability in the plasma for turmeric and curcuminoids. So here you can see that when Term Extra was dosed in 150 milligram of curcuminoids in a 250 milligram dose, and compared with 1500 milligram of the standard 95% extract, the kinetics were equivalent. So at a 10-time lower dose, Term Extra showed equivalent kinetics to the standard 95% extract. And with this, then we conducted a clinical trial in the chronic knee joint pain, where we got results in the improvement in the 80-meter fast-paced walk test and 9-step stair climb test, etc. And then we conducted a second study in the delayed onset muscle soreness. Basically, we wanted to evaluate Term Extra's efficacy in musculoskeletal health. And in delayed onset muscle soreness, there was a significant reduction and significantly faster reduction of pain in the Term Extra group as compared to placebo group. And also the soreness, muscle soreness was reduced very significantly in the Term Extra group, which was reflected in the objective biomarker of serum LDH. And the serum LDH levels also were reduced significantly faster in the Term Extra group as compared to placebo. So taking it further, we asked ourselves another question. Now, I think we have heard a lot of times that turmeric extract or curcumin is absorbed much better with the help of piperine, right? So we asked ourselves a question, how will Term Extra perform if it is compared with turmeric plus piperine, a standard 95% extract plus piperine, and how the blood picture will be. So we conducted a third pharmacokinetic study, and interestingly, Term Extra again in the dose of 250 milligram showed equivalent kinetics to 3 doses of standard 95% extract plus 5 milligram piperine, which was given 8 hourly. So with 3 doses, this was a reflection of a real world dose. In real world, people don't take 1500 milligram in a single dose, right? They take it like morning, afternoon, evening. So we mimicked the same dose of 95% extract. We added piperine, and we compared it with just a single dose of Term Extra, and the results were same, the equivalent kinetics in such a small dose. Of course, very interestingly, piperine addition had no effect on the pharmacokinetics of the standard 95% extract, and I call it a path-breaking study. So taking it further, we conducted the clinical trial in osteoarthritis patients, and here Term Extra started showing significant reduction in pain and improvement in the knee quality of life as evaluated over a Coos validated questionnaire, right, as early as day 7. And at the end of the treatment period, we could see a significant reduction in all the inflammatory biomarkers like interleukin-6, TNF-alpha, HSCRP, and interleukin-1 beta, which showed that term extra could actually arrest the inflammatory process, which is secondary to the degenerative process in the osteoarthritis patients. Let me take you all back to Ayurveda. So Ayurveda praises turmeric as a skin, herb which works great for skin. It improves skin luster, it improves skin texture, and it also acts as a wound healing agent. So with the keyword curcumin, the results are like 25,000 on PubMed. When we added the keyword skin to it, the number drastically dropped to 1,000. And when we tried to evaluate if anybody has tried an internal supplement to get the results on facial skin health, there was no study available. Again, we took up the challenge, and we conducted a clinical trial of Term Extra Glow in improving the skin health in the otherwise healthy women subjects. And as you can see here, the blemish which was selected at the beginning of the trial showed a significant reduction. Term Extra, interestingly, reduced transepidermal water loss right from day 15, and which resulted in the increased hydration and increased skin gloss at the end of the treatment period. So this is our story for Term Extra, both Prolanza and Term Extra. We took the cue from reverse pharmacology. We asked ourselves the research question, what better can we do while retaining the health benefits for these both herbal, botanical ingredients? And we use the entire package of translational research, right from in vitro to in vivo to human pharmacokinetics to human clinical trials, and even for Term Extra, even for the patient population. And we completed the translational research package with the basic insight from reverse pharmacology, and we completed, this is the story of the two products, and of course the research goes on, and the trials are going on for both the ingredients even currently. So I would like to end my presentation with this one verse, which is from Sushruta Samhita. I don't know. Sushruta was a global pioneer of ancient surgery. And I think in the London School of Surgery also his idol is there. So that's the way to pay homage to these ancient Ayurveda scholars. And as he says, study of only any one science is never complete. So a wise physician must be curious and always study from multiple disciplines and use the knowledge and wisdom with discernment. So it is this curiosity which is at heart of the Nutriventia team. We always ask each other a lot of tough questions, then we put our heads together and we try to innovate and get innovative products like Term Extra and Prolanza for users and consumers. I thank CRN for giving us this opportunity to talk to all highly knowledgeable and experienced people, audience like you all. Thank you very much for a patient hearing. Thank you.